Hi, I'm Allison Burke from the Good Food Institute. GFI is a team of scientists, entrepreneurs, lawyers, and corporate engagement specialists who are focused on using markets and food technology to create a sustainable, healthy, and just food system. You can check us out and learn more at gfi.org. In the agenda for this lecture, we will discuss how much meat we are eating, the history of plant-based meat, extrusion techniques for creating plant-based meat, the Couette cell or shear cell technology, sources of plant protein, binders in plant-based meat burgers, mycoprotein, algal protein, and emerging sources of non-animal protein, and the growing demand for plant-based protein in general. To determine what plant-based meat products will be eaten by which populations, it's worth looking at where certain foods, including animal meat, are eaten the most. Where you live influences what you eat, and our diets vary across the world. At 12% of the average adult's daily calories, the Vietnamese diet has the highest proportion of pork. Poultry consumption is highest in Hong Kong at 8%, seafood consumption is highest in Japan at 5%, and beef consumption is highest in Argentina at 11% of the average adult's daily calories. 8% of a German's daily calories come from animal fats, and 22% of a Somalian's daily calories come from milk. Wheat makes up 38% of the daily caloric intake in Libya, and at 7% of their daily calories, Cubans have the highest consumption of starchy root vegetables. North Korea leads the way in consumption of pulses, like lentils and chickpeas, at 6% of the average daily calories, and the Chinese receive 7% of their daily calories from green vegetables. Focusing in on meat in particular, though, we all start to look the same. Even predominantly vegetarian India has seen a 363% increase in the total tons of meat consumed between 1961 and 2011, and globally the trend is the same almost everywhere. Despite regional differences in dishes and ingredients, almost everyone is eating more meat in total and in terms of grams per person, and everyone is eating almost double the number of calories of meat that their grandparents did. Eating more and more meat every year is difficult to sustain. For our waistlines, for the planet's grazable land and feed crop production, for greenhouse gas emissions associated with growing animals and industrial processing them, and for affordability. Luckily, products that substitute for meat are becoming more popular as well, and demand for meat substitutes is growing globally, in some of the countries with the highest per capita consumption of meat. Meat substitutes fall into two general categories. One category is plant-based meat, which includes traditional products like tofu and seitan, and new products specifically engineered to mimic the taste, mouthfeel, and in some cases the nutritional profile of meat. The second category, which is in the very early stages of development, is clean meat, which refers to meat grown from animal cells but without the slaughter of an animal. Instead, clean meat uses techniques borrowed from biochemistry and medicine to culture and grow cells into familiar shapes and textures. In this course, we'll first focus on the techniques and tools used to make plant-based meat, and then provide an introduction to the science and innovation behind clean meat. What is plant-based meat? Any substitute for animal-based meat that is made from plants or fungi can be considered plant-based meat. Usually, plant-based meat products attempt to mimic in taste and appearance a particular animal-based meat product like a chicken breast or ground beef, but this isn't a strict requirement. Products like tofu and seitan can also be used as plant-based meats and can be shaped and cooked in different ways to replace animal-based meat in a variety of dishes. You may be most familiar with veggie burgers or veggie dogs that replace animal meat hot dogs at a picnic, for example. The variety of plant-based meat products and the methods used to produce them have led GFI to develop a mind map that categorizes the inputs and various production processes into protein sourcing, isolation, and functionalization of plant-based proteins, formulation of a plant-based meat product, including the addition of plant-based fats, flavorings, and binders, processing, including texturing, molding, and extrusion, and distribution, including addressing market position, food service, and supermarket consumers. We'll talk through all of these areas of work, and as well as the history of plant-based meat. Plant-based meat products, in which, as you'll recall, we're also including mycoprotein-based products, can be as simple as processing wheat or soybeans to extract protein isolates, but can also involve genetic engineering of plants to produce varieties that express higher levels of protein or subtly improved protein molecules that are easier to digest, mix, or combine with one another or with other nutritive ingredients that would be desirable in a plant-based meat product. The evaluation of plants for their protein contents includes techniques like genomic trait mapping, analyzing the genome of a plant to see which proteins it expresses and what factors improve or detriment the expression of those proteins, as well as digital phenotyping, which can compare multiple varieties or cultivars of a plant before they are even grown to determine which will have better protein yields or grow optimally under the conditions of the environment and territory where they will be produced. Of all the materials used to produce plant proteins, soy is still the most commonly used, with more than 60% of plant-based meat products using soy protein as a protein source. Pea and wheat protein are also commonly used, and food scientists are beginning to explore the potential and uses of other sources of protein, like lupin, lentils, and oats. Up until recently, most plant-based meats were only made from wheat or soy, and now we see other proteins being used and researched, like pea, lupine, mung bean, fungus, and others. A recent GFI intern did a deep dive into underutilized and neglected crops that could provide new sources of plant-based protein, and found several crops that have high levels of protein, low levels of starch, and functional properties favorable to gelation, emulsification, or foam stabilization. In many cases, these crops could supplement or replace soybeans, particularly for products marketed to audiences that want to consume less soy or who are allergic to soy. Other crops were identified as needing further research, but with promising protein profiles and functional properties. Any of these plant protein sources could provide a new ingredient for plant-based meat products. The difficulty is in convincing farmers to grow these crops and convincing ingredient suppliers to invest in processing and providing for sale these new protein sources. Along with identifying new crops that can be used for plant-based meat, crops already in use can be optimized to be better for plant-based meat by increasing the amount or bioavailability of their protein. One research project attempting to do this is being conducted by the Foundation for Food Agriculture and the Open Philanthropy Project. Many pulse species could be better optimized as plant-based meat by characterizing their texturization abilities and the molecular characteristics of their proteins, including where they bind other molecules like flavor agents and how they unfold during the cooking process. These characteristics can be computationally modeled, and genes can be added or edited to pulse seeds to produce varieties with improvements in these characteristics. This research project into developing better pulses for plant-based meat has two phases. In Phase 1, scientists will utilize high-throughput digital phenotyping, genome sequencing, and functional characterization to link genotype, phenotype, environmental conditions, and protein or ingredient functionality to assess the suitability of a variety of pulses for use in plant-based meats. The focus of these efforts is gelation, water-holding capacity, and fat-binding ability, and the structuring ability of the proteins and/or ingredient combinations to form meat-like fibers. This work will yield a publicly available, user-friendly online repository for all the data generated, so that others seeking to develop plant-based meat products will not have to duplicate efforts to generate foundational pulse ingredient functionality and protein structuring data. In Research Phase 2, scientists will use data from Phase 1 to perform precision breeding and editing for improved pulse crops for the explicit purpose of optimizing growth of these crops for plant-based meat ingredients. This would, for example, increase the efficiency of protein isolation from the selected pulse crops, making the process of manufacturing plant-based meats take less time and be less energy intensive. Additionally, there is a need to cultivate crops possessing proteins with desirable functional properties for plant-based meat applications, such as higher cysteine amino acid content to encourage molecular cross-linking, and enhanced fiber formation in downstream processing. Classical breeding techniques may be used to bias the expression profile of plant storage proteins to enrich for accumulation of the subset of the host plant's native proteins that perform best in plant-based meat applications. After choosing a protein source, the protein needs to be isolated, and it may be helpful to fractionate the protein, hydrolyze it, or functionalize it in some way. Protein fractionation is the process of isolating individual types of proteins from a mixture, as many plants are likely to contain multiple types of protein, even at similar molecular weights. Protein hydrolysis is the process of adding water and enzymes and adjusting the pH of a protein mixture to partially digest the protein or change its activity to improve binding properties or improve its ability to form an emulsion. Finally, creating a plant-based meat product requires formulation, including adding flavoring compounds, plant-based fats, binders, and other ingredients that will improve the taste and texture of the product. Because the majority of taste comes from our olfactory sense, flavorings are particularly important if a product is designed to closely mimic an animal-based meat product. We'll first talk more about manufacturing because this process is key to producing the textures we're used to in animal-based meat. Processing techniques commonly used with plant-based proteins include spinning in a shear cell like a Couette cell, molding or folding a protein dough or mixture, extrusion, such as with the twin screw extruder in the top image to the right, or dehydration to produce a tougher, chewier texture. Let's take a step back to 1947 and dive in deeper to what Robert Boyer, the Ford engineer that we talked about in Lecture 1, was trying to do. The launch of technologically driven plant-based meat that could be commercialized started with the production of dry texturized vegetable protein, or TVP, which is produced by the cooking and extrusion of defatted soy meal, soy protein concentrate, or wheat gluten. These products have an elastic and somewhat spongy texture. A further approach used to mimic the fibrous and elastic properties of meat is the production of fibrous raw materials such as shredded TVP and mycoprotein preparations. These raw materials are mixed with water, a binding component, and further ingredients, and are then formed and subsequently steamed. Extrusion as a process heats, compresses, and stretches a material to change its structural alignment and force it into a desired shape. Shown here is a modern screw extruder, but Boyer would have used an equipment like the spinning machine used in 1969 at General Mills, which is shown in the black and white inset. Research on the high-moisture cooking extrusion process at the beginning of the 1990s led to new possibilities for texturing food proteins into distinctive fibrous structures similar to that of muscle. This process produces, in a single step, a fibrous meat-like structure that has a similar moisture level to meat and mimics its mouthfeel. This twin-screw extruder is the upgraded industrial version of the single-screw extruder shown previously, and the inset shows a piece of soy protein produced from twin-screw extrusion with a layered fibrous internal texture. In fact, soy protein can produce a wide variety of textures, shapes, and fibers, with variations in temperature, screw speed, and water content. Like animal protein, soy protein needs to unfold, cross-link, and align to form microscopic and macroscopic fibers. The high-moisture cooking extrusion process, which is characterized by water levels of up to 70%, provides the required processing conditions. Co-rotating twin-screw extruders equipped with long screws and specially adapted long cooling dies are effective at processing a low-viscosity mass of soy protein into a protein strand with a distinctive fibrous structure. In the first step, the ingredients, which are usually protein powders and water, are continuously fed into a long extruder barrel. The co-rotating screws mix the ingredients thoroughly, while the mass is steadily heated to temperatures of 260 to 360 degrees Fahrenheit and moved toward the die section. During the hydrothermal treatment, the proteins unfold and form new covalent intermolecular bonds. Once the mass enters the cooling die section, drag and shear flows align the proteins in the direction of flow. The cooling that occurs in the long die section has several effects. The temperature gradient from the core of the strand to the die wall increases the shear flow, cools the mass to a core temperature below the boiling point of water, and avoids expansion that could be caused by the evaporation of superheated water. As the protein fibers cool, non-covalent hydrogen bonds, electrostatic interactions, and van der Waals interactions develop. The viscosity increases and the mass solidifies to a strand with a meat-like structure. A different method of producing a plant-based meat product with a similar structured fibrous texture is a new device called the Couette cell, or the shear cell, which was developed at the University of Wageningen in the Netherlands. This device operates along a similar principle as an old-fashioned ice cream churn. An inner core with an outer ring rotating around it sets up a pattern of flow that is faster along the edges than around the center core. When protein dough is added to the inner chamber, it is pulled into fibers that align and form structures similar to muscle fibers. Other companies like Nova Meat are looking into 3D printing technology as an alternative to extrusion to produce custom-designed shapes. The ink used is a paste of rice, pea, and seaweed ingredients with the option of adding colors and flavors. Here's one process by which soybeans become textured soy protein and soy protein isolate, which can be added to other products to increase their protein content or blended with other ingredients to make a soy burger patty or other plant-based meat product. First, the raw soybeans themselves are cracked and dehulled to remove their outer casing. In some cases, the hulls can also be used as a food product. The soybean is flaked, from which soybean oil can be extracted, and then either dried, ground, or acid precipitated and separated to form soy flour, soy protein, or a form of soy protein isolate. Similarly, wheat is often made into wheat protein or wheat gluten in a process that involves milling wheat grains to form flour, adding water to make a dough, separating gluten through washing and centrifugation, drying the separated gluten, and producing isolated protein products through extrusion, alkaline precipitation, or enzymatic hydrolysis. The processing of meat analogs in cooking or heat-based extruders involves a multitude of machine and process parameters. The composition of the matrix, the variety of ingredients, and the water content all affect the final product. However, data about the individual effects of each parameter and their interactions on fiber formation and final product quality are scarce and are primarily focused on soy, wheat, and pea protein. High protein levels and cross-linking capability are two of the most important parameters, and it's worth talking more about how proteins cross-link, as well as about transglutaminase, one of the most important enzymes used to link proteins, both plant and animal, to other proteins, and which allows for the creation of Frankenmeat products like the checkerboard sausage shown here. Transglutaminase is a naturally occurring enzyme in plants, animals, and bacteria. Enzymes are proteins that act as catalysts in chemical reactions. They speed up reactions and make reactions occur that otherwise wouldn't. Although transglutaminase is a fairly new product, enzymes have been used for hundreds, if not thousands of years as meat tenderizers and to make cheese. Enzymes that break down starches into sugar are used to brew beer, and naturally occurring enzymes in meat tenderize dry-aged steaks and give flavor to cured meats. Transglutaminase creates a covalent bond between protein molecules by linking the amino acids glutamine and lysine. All proteins are composed of amino acids, small molecules consisting of an amino group, a carboxylic acid group, and a side chain bound to a central carbon atom. Amino acids can be linked together by joining the carboxylic acid of one amino acid to the amino group of a second, but the way transglutaminase binds amino acids is by binding the side chains of glutamine and lysine together directly. Because transglutaminase is an enzyme, this is an enzymatic reaction, which means it can be interrupted by heat, which causes the enzyme to denature or lose its shape. Transglutaminase is stable up to about 100 degrees Fahrenheit, above which it starts to inactivate. Along with being an enzymatic binder that lets us stick proteins together to make new shapes and textures, transglutaminase is also a great example of a cellularly engineered product. The first transglutaminase was produced in the 1960s from guinea pig liver and required the addition of calcium to function. In 1989, a Japanese company discovered that a strain of soil bacteria could be used to produce large amounts of transglutaminase that didn't require calcium to function. This is called microbial transglutaminase, and it's what's used in kitchens and food production facilities today. Even though it's produced by soil bacteria, and we wouldn't normally rub our plant proteins in dirt and eat them, transglutaminase is harmless to eat and doesn't leave any residual flavors in foods. Transglutaminase is given a classification by the United States Food and Drug Administration called GRAS, which doesn't refer to the soil it comes from, but instead stands for Generally Recognized as Safe. We'll talk more later about what it means to classify ingredients as safe and what testing food products need to go through before they can be introduced to the market. While transglutaminase can bind any protein, animal or vegetable, it's not always the right choice for every application, and sometimes the bond desired is a little looser than that produced by transglutaminase, or the processing steps required need a different activation mechanism. To produce a product like a plant-based burger, a little bit of binder goes a long way, but some is necessary to keep the product's overall shape and consistency through the cooking process. Otherwise, you'd end up with a sloppy Joe. Binders are usually gummy or sticky, like cornstarch, flour and water, or tapioca. Starch, gum arabic, xanthan gum, and cognac gum are commonly used, and soy protein isolate himself can be used as a binder, as you saw in the variety of shapes and textures produced by the extruder. Soy protein is happy to bind to itself as well as other ingredients. Binders are the last step we need to make a plant-based burger or nugget. We've isolated plant protein, textured it, and now we know how to make it adhere to itself and to other components like flavorings and seasonings. The last two plant-based meat products we talked about in the timeline of plant-based product development in the first lecture are the Beyond Burger and the Impossible Burger. Though they're made of familiar vegetable and grain proteins, primarily pea and wheat respectively, and coconut and canola oils, they also contain some interesting new ingredients. The perhaps more traditional Beyond Burger bleeds due to beet juice extract, which provides a red color and uses two different oils to mimic the texture of animal fat. The fat in a beef burger is actually multiple types of fatty acids with different melting points, which is why animal meat can be juicy and squishy at the same time. Some of the fat remains solid even after it is cooked, while some of the fat melts. Plant-based fats usually have lower melting points than animal fats, so encapsulation techniques or the use of multiple types of oil is an attempt to replicate this property of animal fat. The Impossible Burger uses two particularly interesting ingredients. One is cognac gum, which is an extract from the root of the plant and is used to provide a gelatin-like texture, as it is usually tasteless on its own. The other ingredient, the result of years of engineering, is soy leg hemoglobin. The soy leg hemoglobin used in the Impossible Burger is actually the product of cellular agriculture, and it is produced by a strain of yeast that has been genetically engineered to produce large quantities of the protein. In general, soy-like hemoglobin is a protein found in the root nodules of the soy plant, where it is produced by bacteria and gives the plant root interiors a reddish color. Like hemoglobin is a globin, which means it is a globe-shaped protein found in both animals and plants. The most easily recognized globin is hemoglobin, which you might know is found in your blood and transports oxygen from your lungs to the rest of your body's tissues through the circulatory system. Hemoglobin also gives blood its reddish color, and as you can see, soy-like hemoglobin is also red. Like hemoglobin is a plant globin, and soy-like hemoglobin is the variant found on the root nodules of the soy plant. There, it cooperates with colonies of bacteria to convert nitrogen into forms that the plant can use as food. Anything left over when the plant dies goes back into the soil where other plants can use it. That's why nitrogen-fixing plants like soy and alfalfa are used in crop rotation schemes. They increase the available nitrogen in the soil. The active ingredient of globins is a molecule called heme, which sits wrapped up in the amino acid chain of the globin. When you heat up an Impossible Burger, the leg hemoglobin releases its heme, the same way myoglobin in animal meat would release heme. Iron-rich heme is part of the characteristic flavor of meat. Later, we'll look more at the flavors that make us believe we're eating meat. Another source of protein that is beginning to be explored is microalgae. Companies like Algama are using microalgae as a functional ingredient that provides protein and vitamins, and incorporating algal protein into products like a vegan mayonnaise and an egg replacement prototype. Mycoprotein is protein extracted from mushrooms or other fungi, so we can't really call it plant protein because fungi are distinct from plants, but it serves a similar purpose in products that need an added non-animal source of protein that doesn't adversely affect taste. Mycotech is one company making mycoprotein and has raised a significant amount of venture capital for its products. One of the largest companies using mycoprotein in its products is UK-based Quorn, which is a relatively high-growth company selling mycoprotein-based products in 22 countries. It is less popular than other plant-based meat companies in the U.S., but its reach is broad enough that most grocery stores carry Quorn products. There are other sources of plant-based protein that require relatively little processing, like king oyster mushrooms, which already have an umami-filled taste and a texture similar to ground meat or pulled pork when cooked. These mushrooms can be directly added to sauces and dishes that would otherwise contain ground meat. As we'll see in subsequent lectures, plant-based demand is driven by the same factors that drive demand for any food product: taste, price, and convenience. Health and environmental concerns are emerging drivers, particularly for younger consumers, but taste, price, and convenience will always carry the majority of a consumer's decision-making. Google's global program chef, who has been instrumental in bringing plant-based products to Google's cafeterias, underscores the point that flavor will be more important to an in-the-moment food choice than health claims. Consumers often state that they care about things like health and wellness, safety, social impact, and others on surveys. However, what consistently affects consumer behavior are the traditional drivers of price, taste, and convenience. In order to displace meat consumption, we need to provide consumers with options that are on par with meat in terms of taste, price, and convenience, the three things that everybody bases their food buying decisions on. Taste is a fundamental driver. We want to eat things that taste good and won't eat things that don't. The fastest growing plant-based brands like Morningstar Farms and Gardein are emphasizing flavor in their products. Another fundamental driver is convenience. Plant-based foods need to be available in the grocery stores and restaurants where the majority of people make food purchases. Having these products available in the main sections of menus and on store shelves next to animal-based products is also key to their adoption. Finally, price is important in getting more people to choose plant-based meat products. The average prices of plant-based meat, cheese, milk, and yogurt are still higher than those of similar animal-based products. Growth in the purchase and consumption of plant-based products is coming from mainstream consumers, not just vegans and vegetarians. Companies like Beyond Meat started with early adopters, but successfully have crossed into the early adopters and early majority customer segments. Older plant-based brands, including Nuttos and Protos, which we learned about earlier, struggled with taste and perhaps with convenience and only targeted a small niche of customers, those with strongly held ethical or religious beliefs that prevented them from eating meat. Today's high-growth plant-based companies are targeting mainstream consumers. From a landscape where only a few religiously motivated companies were producing plant-based meat products, to today's landscape where all the companies creating plant-based protein and those incubating, supporting, and funding those products would take several minutes just to name, methods of producing plant-based protein have come a long way. In this lecture, we learned about the growing global demand for protein, the history of plant-based meat products, methods like extrusion, the Couette cell, and mechanical processing that are used to produce protein powders and plant-based meat pieces with fibrous textures, and we took a dive into the biology behind some of the novel ingredients in the Impossible Burger and in binders used to adhere plant-based proteins together. The takeaways from this lecture are that plant-based meats have a long history, from religious groups who wanted vegetarian alternatives to traditional meals like pot roast and meatloaf, through today's highly engineered plant-based burgers that bleed like animal meat. The process of creating a plant-based meat product involves protein sourcing, isolation, functionalization, formulation, processing, and distribution. New materials, including fungi, algae, and crops not currently used as plant protein sources, will expand the market for plant-based protein and the types of ingredients available. If you found this lecture to be of interest, you can find out much more at GFI.